I think I'm going to get started now. First, I wanted to thank Tyler for setting all this up and doing the logistics. And just as also logistics, if you could mute yourself during the talk, later on as we have questions and discussion, you can certainly unmute yourself as we go through that. And also to preserve the bandwidth, it'd probably be a good idea just to turn off your video, except for our speaker. And so before I introduce today's speaker, just to introduce myself, I'm Ed Levin, Director of the Center on Addiction and Behavior Change, part of DIBS. And thanks to all the folks at DIBS for helping put this on. And I just want to look ahead a little bit. Next Friday, not on Wednesday, but Friday, is September 25th, we're going to have our symposium, and that's together with Durham Tri, which is a community action group. Wanda Boone is the director of that, founder and director, and they've done an outstanding job in the community in terms of trying to prevent substance abuse, particularly among adolescents. So the title is Substance Use Prevention with Equity, a Community and Neuroscience-Based Approach. So we're pulling together academics and clinicians and researchers and with community action people who are actually making enormous differences in our community. So it's really a very important confluence of our efforts. So I hope you can make that. You do need to register, and the registration I think is probably in the chat box or on the DIB site at least. And then after that, on October 7th, we have Walter Scott, Senate Armstrong from our philosophy department talking about does addiction remove responsibility? And so there, in terms of free will and other concepts and how they actually make a real difference in terms of how we treat addiction. And then we're going to go on again with Christine Tu later in the semester. Sandra Comer is coming from, no, not coming from, excuse me, will be zooming in from Columbia, talking about medications development and opiate disorder. And then finally on November 18th, Roy Stein, who is a long-term researcher and clinician from Duke, now at the University of North Carolina, Chapel Hill, talking about his work with opiate addiction. Now I'm putting together the seminar series for the spring, and if anyone has any suggestions or would like to give talk themselves, please email me at edlevin@duke.edu, and I'll be working putting that together over the next few weeks. So now it's my privilege to introduce Dr. Lauren Slosky, who is postdoctoral research fellow in the Department of Cell Biology. And Lauren earned her PhD in medical pharmacology and neuroscience at the University of Arizona and has been here since 2015. And she has done some really very important work in terms of the neurotensin receptor mediated treatments. And today is going to speak to us about leveraging functional selectivity in the neurotensin receptor 1 mediated treatment of addiction. Lauren?
Awesome. Ed, thank you so much for the introduction and for the opportunity to speak today, and Tyler for doing so much of this coordination. I'm happy to have the opportunity to talk about, like you said, our work on the neurotensin receptor 1, and specifically a new class of compounds we've identified that seems to be able to separate on-target therapeutic effects from on-target side effects. This compound, or this class, was developed as part of an ongoing search to identify novel therapeutics for the treatment of psychostimulant addictions. I want to start by introducing addiction, not as necessary in this particular context, I'm sorry, as a brain disorder that is currently causing a public health crisis. In 2016, drug overdose became the #1 cause of death of Americans under the age of 50, and unfortunately, conditions haven't significantly improved in the last 4 years, with preliminary data suggesting that 2019 will see a record number of these deaths. While opioids are primarily to blame for this sharp increase, particularly in the last several years. Abuse of the psychostimulants cocaine and methamphetamine are also on the rise, shown here in this maroon and orange color, and they also contribute to this mortality statistic. As we'll talk about in greater detail in a few moments, the neurotensin receptor 1 is a promising anti-addiction drug target. And it's the desire to target this receptor that's led to the development of a prototypical small molecule I want to talk to you about today, which we're calling SBI-553. I'll show data to suggest that SBI is an allosteric and beta-arrestin biased ligand, and that, like these unbiased peptide agonists, it attenuates the effects of psychostimulants in mouse models of drug use and misuse. However, unlike these peptide agonists, this biased agonist does not produce other features associated with classic NTR1 agonism, things like hypothermia, hypotension, and motor coordination impairment. Finally, I'll show some data to suggest that the ability of SBI to modulate locomotor activity in response to stimulant administration requires beta-arrestin 2, and specifically the expression of this protein by a group of neurons that reside in the striatum and also express the dopamine receptor D2. Why we're particularly excited about this set of data is this. They suggest that the neurotensin receptor 1, this G protein coupled receptor that acts as an endogenous modulator of brain dopamine signaling, but also mediates various other physiological effects, may be one of a growing number of GPCRs for which it's now known that the G protein and beta-arrestin signaling arms mediate distinct physiological effects, and that the selective activation of beta-arrestin downstream of this receptor may actually be preferable to classic receptor agonism because you get this desired attenuation of addiction-associated behaviors without the side effects produced by a more balanced receptor agonist. So this is where we're going this morning. But where I want to start is back with our conversation about addiction. And I just want to say briefly that when we talk about addiction, we're really speaking about the most severe form of a substance use disorder. This is a chronic, relapsing disorder of the brain that is currently understood as a cycle. Binge and intoxication lead to withdrawal and an associated negative affect. This, in turn, precipitates preoccupation, anticipation, we often hear craving, which gives rise to another instance of binge and intoxication. Today, I want to focus on a potential strategy for breaking this cycle of addiction that hinges on our ability to attenuate the acute effects of psychostimulants. These are effects that promote binge and intoxication, but ultimately lay the groundwork for later withdrawal, craving, and relapse. As is often discussed in this forum, substance misuse is marked by changes in how individuals anticipate and respond to rewards. And at the level of the brain, this is mediated by changes in the mesolimbic dopamine system. The central circuit in this system is shown here in the rodent brain. Rewarding stimuli increase dopamine in a region of the ventral striatum known as the nucleus accumbens, shown here, with the source of this dopamine being midbrain ventral tegmental area neurons. This system for reinforcing natural rewards is essentially hijacked by drugs of abuse, which have all been shown to increase dopamine in the shell of this nucleus accumbens structure. And cocaine and methamphetamine, which we'll focus the majority of our time on today, are no exceptions. Unfortunately, direct blockade of this aberrant and in some cases excessive dopamine release isn't a viable therapeutic strategy because of the many diverse roles dopamine plays in the brain. In addition to reward, we're thinking about things like movement and mood and executive function. An alternative strategy is to more selectively reduce downstream dopamine signaling in this circuit. And there's a growing body of work to suggest that this can be achieved by targeting the neurotensin system. Neurotensin is a 13 amino acid peptide, shown here, originally isolated from the bovine hypothalamus, and it acts as an endogenous regulator of brain dopamine signaling. Neurotensin is highly expressed in dopamine-rich brain regions, including the VTA, where it is actually co-released with dopamine into the nucleus accumbens. Neurotensin acts at a high-affinity G-protein coupled receptor known as neurotensin receptor 1, or NTSR1. We'll see this a lot. NTSR1 is also expressed in dopamine-rich brain regions, including purportedly on resident striatal neurons, as well as on the cell bodies and terminals of dopamine neurons, and the terminals of glutamatergic afferents from the frontal cortex. In many of these locations, the NTSR1 co-localizes with the dopamine receptor D2. If we could take a closer look at what was going on in this receptor-receptor interaction, we would see that neurotensin, through action at NTSR1, acts to antagonize the effects of dopamine at D2. It's been shown to reduce the affinity of dopamine for this receptor and attenuate downstream D2 signaling. This antagonistic relationship is thought to underlie its ability to modulate dopamine signaling and have therapeutic value. In fact, when you administer peptide NTSR1 agonists, you see effects that almost directly oppose those seen with the acute administration of things like methamphetamine and cocaine. For example, these peptide agonists attenuate stimulant-induced hyperlocomotion, as well as act as neuroprotectants, antipsychotics, and cognitive enhancers. For all of these reasons, the identification of small molecule drug-like NTSR1 agonists have been highly sought after. Unfortunately, decades of screening using G-protein-based approaches have failed to identify viable small molecule NTSR1 agonist leads. The classic model of G-protein coupled receptor signaling calls for signaling by families of heterotrimeric G proteins and desensitization by a family of proteins known as beta-arrestins. However, it's now been shown by the Carone group and others that beta-arrestins not only participate in receptor desensitization, but also have critical roles in receptor internalization, trafficking, and signal in their own right. New studies are showing that these pathways can be activated independently through the use of biased or functionally selective ligands, and these ligands are showing us that these pathways actually have distinct physiological effects in vivo. The neurotensin receptor 1 is functionally coupled to the G-alpha-Q pathway predominantly, leading to IP3 generation and ultimately the mobilization of intracellular calcium. Neurotensin acting at this receptor also, however, recruits G-protein coupled receptor kinases, which phosphorylate the receptor tail, leading to beta-arrestin association, receptor internalization, and trafficking through kinase cascades, and signaling through kinase cascades. Because of the historical lack of success identifying small molecule NTSR1 agonists using this G protein signaling arm as the readout, namely calcium mobilization, Dr. Barrick in the Carone group developed a screen to identify NTSR1 activators based on the ability of the activated receptor to recruit beta-arrestin. So In this screen, GFP tagged beta-arrestin 2 translocation is measured by confocal imaging. Here on the right, we're seeing neurotensin-induced beta-arrestin recruitment, and this extent of recruitment can be quantified by measuring objects per unit area in a rather high throughput fashion. Using this screen, a large-scale screening and lead optimization effort was undertaken by the Carone Group in collaboration with the Sanford Burnham Presby's Medical Discovery Institute. And it's this effort that's led to the development of the current orally available and brain-penetrant lead, a compound, again, we're calling SBI-553. When I joined the Carone Group in late 2015, we devised a three-pronged approach to understanding the actions of SBI-553. It was newly developed. We wanted to know how it was acting at the receptor, what the consequences of this action were for the cell, what physiological effects it had in animals, and to characterize its behavioral effects in mouse models of stimulant misuse. And so we started with this question of how does this small molecule interact with the receptor, and what are the consequences of this interaction for the cell? And this has been a journey, but I can tell you that we now know that SBI is an allosteric ligand, meaning that while the endogenous peptide neurotensin binds the receptor at what is, by definition, the orthosteric site, SBI binds the receptor elsewhere at a so-called allosteric site. The first evidence of this came from radioligand binding data, where rather than seeing competition, we saw an enhancement of orthosteric ligand binding. What you're seeing here is a plot of specific radio-labeled neurotensin binding in the presence of increasing concentrations of SBI, represented by increasing saturation of this blue color. Here we're looking at increasing concentrations of the radioligand, and we're looking at specific binding in the black with the radioligand alone. Rather than seeing this curve move to the right and down if SBI was competing to occupy the same physical space on the receptor, we see a leftward shift to this curve, or a change in the KD indicating an increase in affinity, and we see an upward movement to the top indicating an increased total number of binding sites available for neurotensin in these membrane preps. So together, these characteristics suggest that SBI is acting as a positive allosteric modulator, binding elsewhere on the receptor, increasing neurotensin's affinity and its ability to bind the receptor. Thanks to a collaboration with Brian Roth's group at the University of North Carolina, Chapel Hill, we no longer have to speculate as to the site of this binding. Brian Crum and this group recently succeeded in crystallizing the rat neurotensin receptor 1, shown here in green, co-occupied by, shown here in red, this neurotensin peptide fragment, the active fragment here in the orthosteric binding pocket, and SBI, shown here in blue, in a novel allosteric site that's defined by transmembranes 1, 2, and 7, and oriented towards that extracellular surface. So, in addition to being an allosteric ligand, SBI has some unique signaling properties. When we evaluated SBI signaling, we looked at it as well as neurotensin peptides and antagonists in multiple measures of both G protein pathway activation and beta-arrestin pathway activation. Because findings within these categories were remarkably consistent, I'm going to focus, or I want to show you right now, the data for calcium mobilization as a readout of G protein activation and beta-arrestin recruitment to the receptor as a measure of beta-arrestin pathway activation. What we see in these assays is that neurotensin itself is a balanced agonist. Balanced in the sense that it both activates G-alpha-Q, leading to calcium mobilization, shown here, and it induces beta-arrestin translocation to the receptor, shown here. Curiously, in these assays, SBI exclusively activates beta-arrestin, such that even at the high concentrations we evaluated, we could not produce calcium mobilization, but found that SBI is as efficacious, if less potent, than neurotensin at inducing beta-arrestin translocation. This is highly unusual and interesting because it suggests that the chemical scaffold of SBI and related family members isn't a scaffold that was simply missed in these 20 and 30 years of screening looking for activators based on calcium mobilization. And indeed, it would have continued to go unnoticed if not for using a beta-arrestin-based screening platform. And so things got a little bit more weird from here. So we have this selective activation of beta-arrestin. But what we also saw is it appears to be able to confer this bias to the endogenous ligand. What I mean by that is that in the presence of SBI, neurotensin-induced calcium mobilization is antagonized. At the same time, neurotensin-induced beta-arrestin translocation is enhanced. This occurs to such an extent that at these higher, low micromolar concentrations, SBI converts neurotensin into a fully beta-arrestin biased ligand. Again, interesting, and interesting from a standpoint where you can imagine with a competitive antagonist competing for the same physical space on the receptor, how you could get broad antagonism of multiple pathways, but this pathway specificity suggests that SBI is selectively stabilizing a receptor conformation that permits beta-arrestin translocation, but is incompatible with G protein signaling. Fascinating, fascinating, but it left us asking, has this allosteric and beta-arrestin biased compound retained any of the positive effects that sent us on this search for neurotensin ligands in the first place? And in particular, we were interested in, does SBI modulate brain dopamine signaling? So advances in small animal neuroimaging are providing non-invasive platforms for assessing central target engagement and biomarkers of this engagement. Here, we're using high-resolution micro-positron emission tomography paired with the radiolabeled glucose probe 18F-FDG. We elected to do this because in addition to having characteristic effects on animal behavior, stimulants also exert characteristic effects on regional brain glucose utilization. This is a study from Dr. Nora Volkow's group in 2008 that partially inspired this work, where they acutely administered cocaine and saw reductions in FTG uptake in a number of regions. Regions that included the striatum, the hippocampus, the motor cortex, the thalamus, among others. And it had us wondering, can we see SBI-induced changes in the brain's metabolic response to dopamine? To address this question, we used a model of L-DOPA-Induced hyperlocomotion. This is a model that allows for much larger differences in locomotor response, and we hoped brain glucose utilization, than achieved with moderate doses of things like cocaine and methamphetamine. In this model, animals lacking the dopamine transporter, these DAT knockout mice, are further depleted of dopamine for signaling by the administration of a tyrosine hydroxylase inhibitor, here this AMPT. Without the ability to synthesize dopamine or to reuptake it from extracellular spaces because they lack the transporter, volitional movement in these animals is essentially abolished. Here you can see we're hugging this zero baseline. However, you can rapidly restore movement by administering the dopamine precursor L-DOPA. Shown here, this is L-DOPA with vehicle in red. When you co-administer this L-DOPA with SBI, you get a reduction in the animal's locomotor response to dopamine. And so these are the groups that we elected to image. Dopamine-depleted dopamine transporter knockout mice, given an acute administration of L-DOPA without, or with SBI-553. PET scans are increasingly read alongside CT or MRI scans, with the combination giving both anatomic and metabolic information. We co-registered our CT scans with a three-dimensional Waxholm-Sprace brain atlas to localize the signal that we're seeing with PET to specific regions. We then use statistical parametric mapping to look for clusters of voxels significantly changed in our different treatment conditions. And what we found was that SBI can indeed alter the brain's metabolic response to dopamine. We're looking first at what happens when you give that bolus of L-DOPA to dopamine-depleted animals. And we found that in the presence of vehicle, we see this expansive cluster of reduced FDG uptake that contacts many of the regions identified by the Volkow group in 2008. Regions like the thalamus, the striatum, and the frontal cortex. However, in animals that received co-treatment with SBI, we failed to identify any clusters of either increased or decreased brain glucose utilization. And if we did a direct comparison of this L-DOPA plus SBI versus the L-DOPA plus vehicle group, we identified a large symmetrical cluster of increased FDG uptake. This large cluster encompassed areas of the striatum, the thalamus, the hippocampus, the midbrain, and the pons. Many of the regions, again, associated with the response to cocaine. And in this case, we have movements induced by SBI in the opposite direction. So with this information, we moved with a little bit more confidence into other behavioral assays, having seen that we are getting a change in how the brain is responding to an acute dopamine bolus. And we moved to something with marginally more face validity, which is stimulant-induced hyperlocomotion. A characteristic of acute cocaine and methamphetamine administration in rodents is an increase in activity that you can monitor in an open field, which is what we're doing here. We're looking at time on the x-axis and total distance traveled on the y. After 30-minute acclimation periods indicated by these gray boxes, animals were given either methamphetamine, cocaine, or saline, and we did that in the presence and absence of SBI. What we see is that the hyperlocomotion induced by both cocaine and methamphetamine was significantly reduced by a 12 milligram per kilogram dose of SBI, but we failed to identify an effective SBI treatment in non-stimulant exposed mice. So this was one feature of unbiased peptide NTR1 ligands that again appears to be retained in this biased agonist. An observation that we made early on about this locomotor effect was the fact that it appears to be beta-arrestin 2 dependent, such that it is lost in global beta-arrestin 2 knockout mice. Here again at the top, we're looking at intact animals where we again see this reduction from methamphetamine and cocaine-induced locomotor activation with SBI treatment. In the global knockouts, however, we fail to see an SBI treatment effect at this 12 milligram per kilogram dose. Our line separation here is lost. And so that was curious, and I'll pick back up on this in a minute, but we moved forward thinking that we were seeing a beta-arrestin dependent reduction in locomotor activity. Because open field activity is only a slight improvement over our anesthetized mice in our PET scanners, we moved to an intravenous cocaine self-administration model in these mice where we could evaluate the effects on self-administered cocaine intake. In this assay, the mice are implanted with indwelling jugular catheters, and they're trained to lever respond for infusions of cocaine, and that cocaine is paired with a Q light. We initially evaluated the effects of SBI using a 3-day paradigm, where animals went in the chamber on the first day after vehicle administration, on the second day after SBI treatment, and on the third day without intervention. And what we saw at this 0.1 milligram per kilogram per infusion dose of cocaine is that SBI treatment significantly reduced cocaine self-administration, such that animals were taking in only about 20% of what they took in with either vehicle or no treatment. Interestingly, this change in intake occurred at the same time that we saw an increase in latency to initiate responding within the session, indicative of potentially a reduced motivational phenotype. Also noteworthy is that this change in latency and cocaine reinforcements was not associated with any change in lever accuracy, indicating that mouse could still discriminate an active drug delivering lever from an inactive non-drug delivering lever, suggesting that this change in lever pattern responding isn't a result of some kind of cognitive impairment. We went on to evaluate the effects of 3 doses of SBI on 4 doses of cocaine. What we saw with vehicle is we have this nice inverted U shape to the cocaine dose response curve, and that this curve is dose dependently depressed in the presence of SBI, such that higher doses of SBI were more effective at reducing self-administration against lower concentrations of cocaine. What was interesting is we also saw that this latency effect was cocaine dose dependent, such that SBI induced longer latencies for less rewarding lower cocaine doses and shorter latencies for more rewarding higher cocaine doses, presenting this interesting idea that this compound, or the efficacy of this compound, is somehow related to the magnitude of the anticipated reward. A finding we found interesting in and of itself, and also because this is a pattern that is also observed with these unbiased peptide NTSR1 ligands. So together, these data suggested that this allosteric and biased compound has retained some of the positive effects of these unbiased peptides, and that we get changes in how the animals respond to the stimulants cocaine and methamphetamine. And it begged the question, can SBI do everything previously attributed to NTR1 activation? We started out asking, is selective beta-arrestin activation sufficient to do anything this receptor has been purported to do? And got to this point where we thought, well, maybe it's sufficient to do everything that this receptor is purported to do. And this is a relevant question because, as I mentioned at the beginning, in addition to modulating brain dopamine signaling, neurotensin also has a role in regulating body temperature, blood pressure, and acute administration has been shown to impair motor coordination. And we wanted to know, can SBI produce these effects as well? As a positive control in these assays, we elected to use a neurotensin peptide analog that's been improved, or that has improved metabolic stability. That's this PD149163 compound. It's been improved to the extent that you could administer it systemically. What we first looked at were changes in body temperature. Here on the left, we're looking at time on the x-axis and core body temperature measured on the y. What we found, as previously reported, is that this peptide NTR1 ligand caused a profound and prolonged reduction in core body temperature. We're talking about a loss of more than 10 degrees that persisted for more than 5 hours at these higher doses. In contrast, we failed to see an effect of SBI even at doses 2.5 times higher than those used in the previous behavioral assays. Additionally, we saw that, again, as reported, this peptide NTR1 agonist impaired the ability of mice to perform a wire hang test of motor coordination. In this assay, mice are suspended by a thin wire by their forepaws, and they're trained to swing their body up and grasp the wire with 2 of their hind limbs. It's a relatively simple task. The mice train on it quickly, and they become very good at it. They do it very quickly. However, administration of this neurotensin peptide dose-dependently impairs the ability of mice to perform this task. This impairment was not seen with SBI, again, even up to this 30 milligram per kilogram dose. Thanks to a collaboration with the Duke Cardiovascular and Physiology Core, we have now been able to look for changes in blood pressure. This is work done by Dennis Abraham and Lan Mao, and what they found was that in animals anesthetized and fitted retroaortically with a pressure conductance catheter, they identified a change in peak systolic pressure with administration of a moderate dose of this neurotensin peptide, but were unable to see a change from vehicle with acute administration of this 12 milligram per kilogram dose of SBI. So these are the data that are underlying this working model, where we have neurotensin receptor 1 joining a group of GPCRs for which it's now known that G protein and beta-arrestin signaling produce distinct physiological effects. If this is true, you may be asking, as we did, SBI blocks in vitro the ability of the receptor to interact with G-alpha-Q proteins and mobilize intracellular calcium and engage any of these pathways associated with G protein signaling. If this is the case, that these effects are mediated by a G protein signaling pathway, can SBI change in vivo how the animals respond to an unbiased peptide? And so that's what we looked at next. In these studies, we took advantage of the fact that in mice, SBI is orally bioavailable. What we did was we delivered either vehicle or SBI 30 minutes before giving an IP systemic administration of the neurotensin peptide. What I'm showing here is the effect of a vehicle-vehicle treatment, and then vehicle in combination with this was a 0.3 milligram per kilogram dose of the neurotensin peptide. Again, we have this profound and prolonged hypothermia. That's what we're looking at here on the left, body temperature. And then on the right, we also see an inactivity phenotype. I haven't talked about this, but you can probably imagine these animals in the presence of the neurotensin peptide, they're hypothermic, they're hypotensive, they have motor impairment. Go figure, they also generally move around less in an open field, and that's what we're looking at here, general horizontal activity. When animals were pre-treated with SBI, however, we saw a dose-dependent blockade both of neurotensin-induced hypothermia, shown here, and this hypoactivity phenotype. To the extent that when we administered a whopping 100 milligram per kilogram dose orally, we could almost completely block neurotensin-induced hypothermia and neurotensin-induced hypoactivity. So these are the data that have built this model where we think G protein and beta-arrestin signaling downstream of the NTR1 produce distinct physiological effects, and that the selective activation of this beta-arrestin signaling arm is not only sufficient to recapitulate the positive effects of these peptides in models of psychostimulant misuse, but it may actually be preferable to classic receptor activation because you get this desired effect without the on-target side effects associated with more balanced receptor activation. So at this point, we were wondering, again, how is this all working? Where is SBI acting on the brain to elicit these changes in purportedly motivated behavior? And in order to ask this question, we wanted to capitalize on the fact that the ability of SBI to block methamphetamine and cocaine-induced hyperlocomotion in the open field is beta-arrestin 2 dependent. To narrow down what neuronal subpopulation is most important for mediating this locomotor reduction, specifically we selected methamphetamine, we took advantage of a series of neuron-selective beta-arrestin 2 knockout mice. These were mice generated by Nikhil Ors earlier in the laboratory. And what they are animals that are selectively missing beta-arrestin 2 in either D1 or D2 receptor expressing neurons. And we elected to look at these two populations initially because they define two largely mutually exclusive groups of cells that have opposite effects on the response to locomotor stimulation. And so what we did was this assay we've seen several times now where animals go into the open field, acclimate for 30 minutes. In this case, everyone got a 2 milligram per kilogram dose of methamphetamine and either vehicle or a 12 milligram per kilogram dose of SBI. And what we see is that in animals missing beta-arrestin 2, solely in D1 expressing neurons, we continue to see a wild type-like response to this biased ligand. However, in animals missing beta-arrestin 2, selectively in D2 receptor expressing neurons, we fail to identify an SBI treatment effect, suggesting that the ability of SBI to modulate methamphetamine-induced hyperlocomotion requires beta-arrestin 2 selectively in D2 neurons. We thought we could do a little bit better because there are multiple populations of D2 receptor expressing neurons in the brain. And again, several of them exert opposing effects on the response to these stimulants. And so we generated another set of mice that were selectively missing beta-arrestin 2 in either midbrain D2 receptor expressing neurons or striatal D2 receptor expressing neurons. And what we found was that in the midbrain D2 beta-arrestin 2 knockout mice, we continued to see some effect of a 12 milligram per kilogram SBI dose, albeit not a wild type response, but some effect of drug treatment. In animals missing beta-arrestin 2 in striatal D2 neurons, the treatment effect of SBI was wholly lost, suggesting that the action of SBI on this methamphetamine-induced hyperlocomotion is not only beta-arrestin dependent, but requires beta-arrestin in these striatal D2 neurons. Whether SBI is acting directly or indirectly on this population is unknown, but it narrows our view to some extent. And it was exciting to us because this is a cell population on which neurotensin peptide itself is thought to act to modulate responses to dopamine. And as a major locus for reward in the brain, we're talking about spanning drug class, the action here could have the potential for broad utility in the treatment of chemical addictions. And given the current climate and the ongoing opioid epidemic, we were interested in what that meant for opioids. This is an ongoing area of investigation, and actually the Sanford Burnham and the Crone group have a joint UG3 UH3 grant from NIDA that is going to look in much greater detail about the interaction between SBI and opioids. But I want to share some of the initial data. Here we're looking at self-administration of the short-acting opioid remifentanil. Here we're looking at different remifentanil doses on the x-axis and the number of self-administered reinforcements on the Y. And what we see is similar to our dose response curve for cocaine. With SBI, we do see some reduction in self-administered remifentanil reinforcements. And again, similar to cocaine, this reduction in self-administration is not concurrent with a change in lever accuracy, suggesting again that the change in lever responding pattern may not be secondary to some kind of cognitive impairment. Much more work needs to be done in this area, but I just wanted to end with a plug for what I see as a need for a pan-addiction therapeutic or classes of therapeutics that can address polysubstance use. You may have seen these kinds of articles, but the concurrent rise of both opioid and stimulant misuse has led to some relatively alarmist titles, both in popular news outlets as well as in journals. Here we're looking at meth in the morning, heroin at night, inside the seesaw struggle of dual addiction. And here they're calling meth and opioid addiction twin epidemics. And while they do seem relatively alarmist, I would argue that given the data, they're accurate. I was rather surprised, although maybe I shouldn't have been, by an article that came out earlier this year that was looking at 16,000 patients moving into treatment for opioid use disorders. And what they found was that in this group of patients, the number of individuals reporting past month use of some other non-opioid drug was astounding. We're talking about around 96% of individuals meeting criteria for opioid use disorder reporting within the last 30 days having used some other substance. Somewhere around 30 to 35% had used within the past month cocaine, and somewhere around 20 to 36% reported using methamphetamine, which is in the last 30 days. I think the prevalence of polysubstance misuse really demands an investigative focus preclinically on substances that are on therapeutics that may be able to span drug class. And so with that, I can go ahead and summarize. We've seen that SBI is an allosteric and beta-arrestin biased ligand. We saw that it blocks L-DOPA-induced changes in regional brain glucose metabolism and that it attenuates psychostimulant-associated behaviors. Today, we looked at locomotion and self-administration. These changes in psychostimulant-associated behaviors have not been associated with changes in or body temperature, blood pressure, or motor coordination, suggesting, if not devoid of these effects, we have produced a therapeutic window where one before may not have existed. Finally, we saw that the ability of SBI to modulate methamphetamine-induced locomotion requires beta-arrestin 2, specifically in this subset of resident striatal neurons. I can just say that we appreciate that the largest clinical obstacle to sustained addiction recovery is relapse after periods of abstinence, and we're currently working to test SBI in those more clinically relevant models, so please stay tuned. We're also interested in understanding more of the mechanism by which this class of compounds is causing these effects in mice, including whether this is occurring through a direct receptor-receptor interaction with the dopamine receptor D2, which we can look at in vitro, and we're also developing tools to assess this in vivo. tools that include conditional NTSR1 knockout mice, and reporter lines that will allow us to visualize NTSR1 expressing cell bodies and projections. One of the future directions I'm most excited about is this incorporation of non-invasive imaging formats like PET-CT with longitudinal self-administration. This is particularly interesting because having a clinically utilized imaging modality will allow us to ask questions like, does chronic self-administration in mice produce the same neurochemical changes characteristic of addiction in humans? Can these changes be corrected with intervention? And also, because with FDG, we can look for broad brain-wide patterns of activity, asking things like, are there central biomarkers of propensity to acquire self-administration, extent of drug intake, or vulnerability to relapse? So again, stay tuned. With that, I would like to thank the large and talented team that has made all of this possible, including Dr. Mark Carone and Dr. Lawrence Barrick, my postdoctoral advisors, their large lab group, which has been extremely helpful, our collaborations with Alexandra Badea in the Department of Radiology, who made analysis of those PET scans possible, collaborators in the Duke Mouse Behavioral and Neuroendocrine Core, where the self-administration work is being done, the Imaging Center, as well as Dr. Roth's group at the UNC Chapel Hill, and then the Duke Cardiovascular Physiology Core and Sanford Burnham for providing these compounds. And with that, I'd be happy to take questions, suggestions.
Well, thank you very much, Lauren. That was comprehensive, just very impressive, elegant studies from a number of perspectives. So we do have a few minutes for some questions. I guess I'll get started. Whenever you give SBI chronically during stimulant self-administration, does it gradually disappear, or might there be the risk of having the emergence of self-administration that would be resistant to this neurotensin mechanism?
That's a fantastic question and something we have given some thought to, although I can't answer that with data at this time. I will say that we know from pharmacokinetic studies that the half-life of SBI is moderate. We're talking somewhere around 3.5 hours, and that when we test animals 24 hours after treatment, we don't see, at least in the self-administration paradigm, a change from their prior treatment levels. But I think the question of repeated administration at high doses is an important question and not something that we had done. We've done these changes between dose levels, but haven't chronically given a 12 or higher milligram per kilogram dose to look for tolerance to the SBI treatment effect. But something I think we absolutely should do.
Okay, thanks. Anybody else have some questions or comments? Remember to unmute yourself if you do.
I've got one. Really nice talk, Lauren. I'm wondering, I love the implications for addiction, but I'm wondering how broad are Brian Roth's group looking at this? Like, I can think of all kinds of illnesses with medications that have side effects, and how broadly applicable do you think this will be?
Yes, absolutely. So I think that the current consensus is that this type of bias signaling is going to be a phenomenon that applies to most GPCRs. Whether they produce entirely distinct physiological effects, I somewhat doubt, but I think the idea that you can selectively engage one pathway or another is something that's been brought to bear on other receptors, things like the dopamine and the beta-2, the mu-opioid receptor as well. They're developing better ligands for that. And so I think that this is going to be somewhat of a general principle, although I think the ability to completely eliminate or retain some features is unlikely to be that expansive. My guess is that these pathways contribute to some combined effects, and we just don't know what those are.
Mm-hmm. That's really exciting. Thank you.
Yeah. Other questions? If you don't speak up, I'll ask another.
I have a quick question. Did you look at the effect of this compound on feeding behavior?
Oh, yes, that's a great question. So neurotensin peptides tend to be anorexic, and we were interested in whether that may be a feature that was retained or lost with this biased ligand. We've done some milk drinking studies. They're drinking either non-fat dry milk or with some concentration of sucrose. And we do see some effects that kind of mirror a cocaine study where the efficacy of SBI is greatest against the less rewarding food product, but really felt like we were getting out of our depth. We have sent the compound to Gina Lininger's group at MSU, and they are pursuing food intake studies in, I think, a more comprehensive way than we could have. So I am anticipating some effects, but waiting to hear what they find those to be.
Thank you.
Any other questions from somebody? I had another one. So how far along is SBI in terms of getting into humans?
So I think everyone is holding their breath. This UG3UH3 grant that I referenced is a milestone-based grant with first-in-human trials anticipated in years 3 or 4. We're currently in year 2, and so unfortunately we have not much to do with the progress there. This is being led by Sanford Burnham and their contract research organization, and I can tell you that the largest obstacle has actually been getting adequate exposure levels in higher order mammals to do those IND-enabling toxicity studies. So SBI is orally available in rodents. It turns out that it is not in higher order mammals. They think it's some kind of GI tract pH issue. So they've spent a year, at least a year now, trying to reformulate and have synthesized backup compounds that may allow for larger oral or larger exposure after oral administration. So stay tuned, but nothing that we'll be doing. We just wait to hear from what they're finding.
It's always good to have backup compounds. So have you thought of, well, I'm sure you've thought of it, you have plans for using SBI against nicotine or alcohol?
Yes, so again, I guess the theme here is get the experts involved. So we have given SBI, or in the process of giving it to Zoe McElligott at UNC to look for changes in alcohol drinking and seeking behaviors. And we actually don't have ongoing projects with nicotine, but I would anticipate if this is indeed an, oh, hi Ed. If this is indeed an NTR1 mediated process, there are reports of NTR1 changing nicotine self-administration. So
yeah, well,
can bring a tube over.
We do tons of nicotine self-administration, so yeah, we should talk about that. That'd be great.
That'd be fantastic, yeah.
Good. Anyone else? Okay. Well, I wanted to thank you so much, Lauren, for such a wonderful presentation and a very promising compound. And it's good that you mentioned there are backups, because it's always good to have backups.
Definitely. Well, thanks again for the opportunity to talk today.
Yeah, that's great. That's great. And a great discussion, too.